All right, this chapter 5, deals with all of those non-Mendelian traits. These are traits that were discovered after Mendel did all of his work. He was lucky enough with the pea plants to be working with traits that had dominant and recessiveness. So 2 alleles for each trait. One was dominant, one was recessive. We could do the Punnett squares. We could figure out the probabilities. Things we're going to talk about in this chapter are kind of change some of those probabilities because it's different types of inheritance. And so the question is, what do all of these have in common? These are all showing pictures of non-Mendelian traits. This one here, this actor Peter Dinklage, who was in Game of Thrones, which my kids always watched. Achondroplasia. Oops. And that's a lethal allele. On your blood types, those are multiple alleles. And then this is flowers showing codominance. So all sorts of other types of inheritance that are considered non-Mendelian. So we're going to look for this first part at lethal alleles, incomplete dominance, I should say codominance, and then kind of contrasting them from regular dominance and then multiple alleles. And this is showing you that when Mendel did his work, again, he just was looking at 2 distinct forms, but we know that there's all these other things that can influence your basic phenotypic ratios. And the first one is lethal alleles. And lethal alleles, like the name suggests, if you inherit a lethal genotype, it's going to cause death before the individual can reproduce. Okay? So it basically removes one of the expected genotypes, okay, that would have been expected of the offspring. And so it drops your ratio down to a 2-to-1 ratio. Remember, Mendel would have had a 3-to-1 ratio, phenotypic ratio. And it's usually a double dose of the dominant allele that's lethal, okay? And the 2 examples are achondroplasia, which is a type of dwarfism, and Mexican hairless dogs. So if you look at Mexican hairless dogs, the having hair is the recessive trait, and being hairless is the dominant trait. So it's a dominant allele. Well, if you inherit the homozygous dominant allele, that's the lethal allele right there. If you're heterozygous, you're Mexican hairless, and if you're homozygous recessive, then you have some hair. This is a Mexican hairless dog. I don't know why one person would have one of those. They look kind of ugly, but that's just my opinion. Okay, so if we cross 2 Mexican hairless, normally you get that 1 to 2-to-1 genotypic ratio, but this group dies as embryos. So then you end up with a 2-to-1, 2 Mexican hairless to 1 hairy ratio. Now, if we just took a hairy dog and crossed that with Mexican hairless, then all offspring would survive, but you'd have about a 50-50 chance of having Mexican hairless dogs. I was going to say, can you have this? Ever have this combination? 2 parents like that? No, because they both would have died already, right? As an embryo. So they wouldn't have survived to adulthood. This also occurs in achondroplasia, a form of dwarfism. The same thing would happen there if 2 individuals who had this type of dwarfism had children together. 1/4 would be taken out because they would die as embryos. So then there'd be a 2-to-1 chance of having achondroplasia versus having normal height. And if a normal height person had children with a person with achondroplasia, then you drop it to, it would be 50-50 chance of normal height children and people with children with achondroplasia. Incomplete dominance means that the heterozygous phenotype is between those of the 2 homozygotes. Okay, so that would be like if in flowers, if you had this being red and this being white, now the heterozygous doesn't take the red color like we would have in normal dominance, but it would have its own phenotype that's in between those two. So it would be pink, okay? So in humans, familial hypercholesterolemia is an example of incomplete dominance, where if you have a heterozygote, then you get kind of a medium amount of receptors for LDL cholesterol, okay? So you're kind of right in the middle. So you get just kind of a medium amount compared to those that are homozygous for it. With multiple alleles, as the name suggests, these are genes, because it's multiple, genes that have more than 2 alleles. So now instead of just big A, little a, there'll be more than that. There'll be more alleles. And so our example is human blood type. Here's one allele, the IA, the IB allele, and then the lowercase I. Okay? The thing to remember is even though there's multiple alleles to choose from, individuals can only carry 2 alleles for each gene. Okay? You get one from mom and one from dad. Okay, so you can still only inherit 2 genes at a time. All they're saying is that there's more than 2 available. Okay, there's more than 2 out there. And so things like PKU and cystic fibrosis actually have lots of different alleles, many alleles that could code for that particular condition. Codominance is when both alleles are expressed equally. So they're both going to show up. So again, thinking with blood types, the IA allele that produces the A antigen on your red blood cells and the IB that produces the B antigen on your red blood cells, they are codominant. So if you inherit both of those, you're going to have type AB blood because you're going to get equally both of them. The lowercase I does not produce any antigens, so there'll be nothing on your red blood cells. The reason I capitalize these I's is because IA and IB are dominant to I, the lowercase I, but they are codominant with each other. So this is what your red blood cells would look like depending on what type of blood type you have. Notice you can either be homozygous for A or heterozygous. Same with B, homozygous or heterozygous. And then this is how we'd write if you had AB blood. And if you had type O blood, that means you inherited 2 of the recessive little i's. You have no antigens on your surface of your red blood cells. So if you look at this, you can see if one parent has type A and one has type B, look at all the different combinations depending on what kind of type A they are. Are they both homozygous? If so, all the kids would be AB. Is mom heterozygous for A and dad homozygous for B? Then you'd have 50-50 chance of AB blood and B blood. If mom's homozygous for A but dad's heterozygous for B, then you have a 50-50 chance of AB blood and type A blood. And if they're both heterozygotes, look at, they could have one of every kind right here. B, A, something, let me go ahead, it's covering it up. Okay, so lots of different combinations. Notice we're still doing the same basic Punnett squares, it's just that there's more than just 2 alleles to choose from. All right.